Hello and good afternoon friends, welcome to the CEC-UGC live lecture. Dear friends, with our ongoing series on development biology, today we would be talking on external fertilization, and for this discussion we have once again with us in our studios Dr. Charu Dogra Rawat. Dr. Charu Dogra Rawat is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. So let's welcome our guest Dr. Charu Dogra Rawat and let's try to grab maximum knowledge through her experience. Hello ma'am, welcome to the EduSet lecture.
Thank you. Good afternoon everyone. So today we will be talking about fertilization, in particular external fertilization. The purpose of any living being is to reproduce, is to produce its own progeny and leave it in the world. So the beginning of a new organism starts essentially by this process which is termed as the fertilization. So the specific objectives for today's session would be to understand the external fertilization, and we'll take the example of sea urchin for this case. We'll understand the action of the sperm and we'll understand the reaction of the egg. So fertilization is a process whereby the sperm and the egg, collectively called the gametes, fuse together to begin the creation of a new individual with its genome derived from both the parents. So the accomplishment of this process of fertilization, there are primarily two. One is that the fusion of the genes from the two parents take place. So it's related to the genetics, it's related to the heredity, that the organism passes on its gene pool to the next generation or to its offspring. And through this process of fertilization, this fusion of the gene or the mixing of the genome from the two parents takes place. The second is the creation of the new organism. So actual formation of the new organism starts when the fertilization occurs, when the two gametes fuse together. There are four major events which are involved in the fertilization. First is the contact and recognition between the gametes, ensuring that they are from species specific. This is very relevant when there is external fertilization. We'll talk about this more. But the first major event is the contact and recognition between the sperm and the egg. The second is the regulation of the sperm entry into the egg. Third is the fusion of the genetic content of the sperm and the egg. And fourth is the activation of egg metabolism to start the development. So the two gametes coming from two different parents need to come in contact with each other, need to recognize that they are species specific. Then there has to be a regulation at the sperm entry, because there is, if it is a monospermic fertilization, then usually only one sperm fertilizes the egg. So there has to be a regulation that only one single sperm enters the egg. Then after the sperm enters into the egg, the first accomplishment of the fertilization process is achieved, which is the fusion of the genes or the mixing of the genome from the two parent organisms. And the last one is the second accomplishment, which is the creation of a new organism. So the activation of the egg metabolism to actually start the development of a new organism. So these are the main four events of the fertilization process. So we today talk about the external fertilization. By external fertilization, we mean when the fusion of gametes takes place outside the body. For example, many marine organisms release their gametes into the environment. This environment can be as small as a tide pool or as large as an ocean. So when the fusion of the gametes is taking place outside the body of an organism, it is termed as the external fertilization. Now, external fertilization faces two difficulties. First is the meeting of the gametes in such dilute concentration. When we are saying that the gametes are released into the external environment, then obviously the environment is too large. For example, if it's a large ocean, then the meeting of the gametes, it's at the stake that the two gametes can come into contact with each other. But when they are open in the environment, it's a very dilute environment which is there. So the first difficulty is the meeting of the gametes, the actual coming of physical contact of the two gametes which is there. Then the second difficulty which external fertilization faces is the prevention of sperm from other species to fertilize egg of a different species. Since the gametes are released into the external environment, it's not necessarily that the gametes lying in the open environment are only from a particular species. There is no restraint, there is no boundary. So, a particular environment can have gametes from many different organisms which are there. So, external fertilization has to counter this difficulty that the fertilization occurs only between the same species, or the fertilization is species specific. So, these two difficulties are faced, which is not there in the internal fertilization when the fertilization occurs inside the body of an organism. So, inside the body of the organism there is a smaller environment, therefore the dilution is not there, the sperm and the egg, they are in the closer proximity, they can be in the closer proximity, and therefore it is a concentrated condition. In second, species specific problem does not exist, because the copulation must have taken place between the same species, and that is why the sperm or the male gamete has entered into the female body. And because the development takes place usually inside the female body, therefore, it is not necessary that now at that stage the species specific thing has to come into play. So, these two difficulties, the mechanisms have evolved to counter these difficulties, which is basically the species specific attraction of the sperm and the species specific sperm activation. So, to counter the meeting of the gametes in such a dilute concentration, there are mechanisms which we will talk about in detail, in which species specific attraction of the sperm takes place. So, some kind of chemicals are released or some kind of cue is there by which the gametes from a particular species only tend to come together. And second is the activation of the sperm, usually because the male gamete is the one which is travelling and the egg and the female gamete usually lies down at one place. Then therefore, there is a process by which the sperm gets activated only when it meets the egg of the same species. So, species specific sperm activation, the mechanisms have evolved that counters this particular difficulties. Now, before we get into the detail of the mechanism of fertilization, I would like to review here the structure of the gametes. This is in particular context with the sea urchin, which we are going to talk about today. So, sperm, as we know, the sperm has this kind of a structure. It has a small head with a long tail. The head is, the major area of the head is occupied by a nucleus which contains the genome, because the primary role of this fertilization is to pass on to the genomic content. So, the major portion of the head is occupied by the nucleus containing a highly condensed genomic material. Then at the tip of the sperm there is a vesicle which is called as the acrosome. This acrosome contains many degradative enzymes. As we will see, they are very much required for the process of fertilization to occur, and these hydrolytic enzymes passes or degrades the the envelope of the eggs, so that the nucleus can enter into the egg and finally fuse with the female's nucleus. So, this is the head region. Then you have the centrioles, they will aid in the formation of the spindles for the mitotic divisions, the cleavage that follows that divisions to take place. There are series of mitochondria which runs halfway in the middle piece in the tail, and this mitochondria is basically that provides the energy for the flagellar movement, so that is why the sperm is motile. And as we have seen that there is always a gamete differentiation in sexually reproducing individuals. The male produces the sperm and female produces the egg. So, egg usually lies at one place, whether it is external fertilization or whether it is internal fertilization, and the sperm has to move and reach to the egg. So, sperm is provided with this flagella for this containing flagellar movement. So, mitochondria provides the ATPs that causes this flagella to move, and you can see that it has a typical 9+2 arrangement of the flagella containing all the dynein arms and all those radial spokes, etc., for the typical whip-like movement of the flagella. So, that is how the sperm looks like. An important feature in these sea urchin sperm is the presence of an actin filament here. So, between the nucleus and the middle piece there is an actin protein which is present in the sea urchin sperm. Now, looking at the egg is contains vitelline envelope. So, there is a plasma membrane for the egg, and then there is an outer layer which is called as the vitelline envelope, and then there is an egg jelly. So there are certain cells, these are basically the substance released by the egg itself and creates a kind of a jelly outside the vitelline membrane. So this vitelline, so this is the egg plasma membrane, then there is a vitelline membrane, and then there is this egg jelly which is there. Just inside in the cytoplasm, in the egg cytoplasm, on the periphery of the egg lies these cortical granules. We will talk about the function of these cortical granules. They again contains hydrolytic enzymes which are there. This is the female pronucleus and this is the female cytoplasm. So egg is much larger in the size. This is the original picture of an unfertilized egg. So you can see that the egg is, now the nucleus is already in the haploid stage, and this is the structure of the egg we are looking at. So this is how the gametes look like. Now to start with the interaction of sperm and egg, it generally proceeds according to 5 basic steps, and these steps are, the first is that the sperm and the egg needs to come together, and this coming together is basically aided by attractants, which are the factors, or there is some kind of a chemical attraction by the soluble molecules secreted by the egg. So egg releases these chemoattractants, by which we will see in detail that by which the sperm gets the cue and it moves in the direction of the egg. It also activates the sperm so that it propels and comes near the egg. So there is an activation of the egg, there is a directional cue given to the sperm and the activation, and it reaches the egg to fuse it. So first aspect is basically the chemoattraction or the chemotaxis, which is called as. The second is the exocytosis of the acrosomal vesicle to release its enzyme. Now when the sperm reaches the egg, before the nuclei fuse, the sperm has to penetrate through the jelly layer and then the vitelline membrane, and then the plasma membrane of the egg and the sperm can fuse. So this penetration is aided by the hydrolytic enzymes which are enclosed in the acrosomal vesicle present on the tip of the sperm, on the head of the sperm. So this acrosomal vesicle, the exocytosis occur and it releases the enzymes in its vicinity, which causes the local degradation of the egg envelopes so that the egg plasma membrane and the sperm plasma membrane can fuse. The next thing is, then when the sperm now is making its path and has finally reached the egg plasma membrane, the binding of the sperm to the extracellular envelope or the vitelline layer or the zona pellucida of the egg occurs. So acrosomal vesicles aids to penetrate the sperm through the jelly layer, and then finally there is a binding of the sperm to this extracellular or the vitelline layer in case of sea urchin and zona pellucida in case of mammalian, when we will see mammalian fertilization. So next process is that. The next step is the passage of the sperm through this extracellular envelope. So now it passes through this vitelline envelope also, and the last is the fusion of the sperm and the egg plasma membrane. So when we are talking just about the interaction, and after that the nucleus of the sperm will pass on to the egg and then the fusion will take place, but the initial interaction, when we are talking about the contact and recognition of the two gametes, so these are the 5 steps that takes place. The steps 2 and 3 are reversed in mammalian fertilization, we will see in our subsequent sessions, and the sperm binds to extracellular matrix of the egg before releasing the acrosomal contents. So this figure clearly depicts all the steps together. So you can see this is yellow is the jelly layer, which is the extracellular coat. This red one is the vitelline envelope, which is extracellular matrix, and this is the egg plasma membrane. So when the sperm is coming, the first sperm contacts the jelly layer. So sperm is contacting the jelly layer. Then the acrosomal reaction will take place, in which the acrosomal vesicle bursts open, releases these hydrolytic enzymes, and these hydrolytic enzymes causes the local degradation of the jelly layer so that the sperm can move further in. And here you can also visualize that this actin has been thrown into an acrosomal process. We will talk about this again in detail. But then this is how the acrosomal process is extending, and these hydrolytic enzymes are also degrading the local jelly layer, and therefore the sperm is moving forward, or it is penetrating this jelly layer. Then after the digestion of the jelly layer, finally there is binding of this acrosomal process to the vitelline membrane. So when the sperm has reached there, the binding occurs at this point, and then this sperm plasma membrane and the egg plasma membrane fuse together in this form, making this kind of a cytoplasmic bridge through which the nucleus and the tail and everything can pass on to the egg, to the inside of the egg. So in this kind of a structure is formed, which is basically called as the fertilization. So this is actually what we are talking about, is the action of the sperm. We are not yet talking about the reaction of the egg. So this picture clearly tells that how the sperm comes in and how it finally contacts the plasma membrane, and how the final fusion of the plasma membrane of the sperm and the egg takes place. So attraction of the sperm, will take these steps into a little detail now. So attraction of the sperm, as we talked about, is basically by the method of chemotaxis. So chemotaxis is when a chemical substance is released into the environment and a gradient is created, and the molecule which are moving follows, or the sperm which is moving follows these chemical gradient and reaches the other point. In many species, sperms are attracted towards eggs of their species by chemotaxis, which is by following a chemical gradient of a chemical secreted by the egg. The mechanism of chemotaxis differ among species, and the chemotactic molecules are different even in closely related species, particularly in external fertilization, where we have already seen that there is a difficulty of species specific fertilization. Therefore, these chemotactic molecules that have evolved are quite different even in closely related species. So one chemoattractant will not attract the sperm from even the closely related species. So that that again emphasizes the species specific fertilization only takes place. The sperm motility is acquired when the sperm are spawned. So this is in particular in case of sea urchin. The sperm are non-motile when they are released into the water, or inside the testes they are non-motile because the sperm cannot move as the internal pH is kept low. So the gonadal pH in case of sea urchin is kept low due to the release of carbon dioxide, and this pH is at this pH the sperm is non-motile. So once the sperms are spawned, the sperm's pH is elevated to about 7.6. That causes the activation of the dynein ATPase and splitting of ATP that provides the energy for the flagellar movement to take place. So dynein ATPase, which is present in the tail of the sperm. So they becomes activated when the pH is elevated. Therefore, the sperms only become motile when they are spawned into the sea water. The direction is provided by small chemotactic peptides. In this case, for example, such as RESACT. There is a protein, a peptide, 14 amino acid peptide, which is isolated from the egg jelly of sea urchin's egg, and this RESACT provides the direction to the sperm. The RESACT diffuses readily in sea water and has profound effect at very low concentration. So RESACT is released by the egg, particularly by egg jelly. So there is a chemical gradient that is created. The concentration of RESACT is much higher near the egg, and then it becomes reduced. The sperm has the receptors for these RESACT molecules, the chemoattractant molecules, and therefore it gives the direction to this to the sperm, that in which direction the egg is lying, and therefore the sperm starts moving in that particular direction. In fact, RESACT performs dual role. Not only does it give the direction, but it also causes the activation of the sperm, because it acts as the sperm activating peptide to move in that direction. So this is basically the model how this sperm chemotaxis in the sea urchin, for example Herbaceae punctulata takes place. The binding of a chemoattractant, which is ligand to the receptor, a membrane bound guanylyl cyclase, activates the synthesis of cGMP from G. So this is basically the sperm plasma membrane, and at the sperm plasma membrane there are these receptors, the GC receptors. The ligand is the RESACT molecule. When the RESACT molecule attaches to this receptor, there is the activation of the synthesis of GMP takes place. This cyclic GMP possibly opens the cyclic nucleotide gated potassium selective channels. So it causes the activation or the opening of this potassium activation channels, thereby causing the hyperpolarization of the membrane. The CGMP signal is terminated by the hydrolysis of the CGMP through phosphodiesterase. So, there is a PDE activity which is present, and therefore, the CGMP is basically inactivated also, causing the inactivation of the GC. Now, since the membrane is now hyperpolarized, the hyperpolarization activated and cyclic nucleotide gated channels allow the influx of the sodium ions that leads to the depolarization and thereby causes a rapid calcium entry through the voltage activated calcium channels. So, this depolarization causes the calcium channels to open, and there is a rush entry of the calcium channels that occur, calcium ions that occur. Calcium ions interact by some unknown mechanisms with the exoneme of the flagellum and causes an increase of the asymmetry of the flagellar bead and eventually a turn or bend in the swimming trajectory. So, wherever the sperm is moving, it causes the bend in that, and therefore, it orients itself towards the direction of the egg. Calcium is removed from the flagellum by the sodium-potassium exchange mechanism. So, this is how the movement now takes place. In fact, this calcium influx also causes the activation of the dynein arm, as we saw. So, it also activates it, and therefore, the sperm, so it not only gives the direction, but also gives the power, the motive force to the sperm for the movement towards the egg that is taking place. Now, this is the part where now the contact or basically the stimulus has been there, where the sperm is now directed towards the egg. Now, when the sperm reaches the egg, it first encounters the jelly layer of the egg. Then what happens is the action of the sperm, also termed as the acrosomal reaction. So, what happened in the acrosomal reaction is that it has two components. One is the fusion of the acrosomal vesicle with the sperm cell membrane, and exocytosis that results in the release of the contents of the acrosomal vesicle and the extension of the acrosomal process. So, here you can see that this is the head of the of the sperm which is shown. This is the nucleus. This, as we talked about, this is the globular actin molecule which is present in the sea urchin sperm, and this is the acrosomal vesicle which is there containing all the enzymes, hydrolytic enzymes such as hyaluronidase, etc. So, this is the acrosomal membrane, and the outside is the sperm cell membrane. So, what happens is that the fusion of these two membranes takes place, and there is an exocytosis of this vesicle, and the enzymes are released into the environment. So, when the enzymes are released, so the portion of the acrosomal membrane just beneath the sperm plasma membrane fuses with the membrane to release the contents of the acrosomal vesicle. So, both these membranes fuse together, and as a result the contents of the acrosomal vesicles are released, causing the local degradation of the jelly layer. Furthermore, as we saw that there is the actin molecules which are there, and actin molecule just polymerize together. Actin molecules polymerize together, and these actin molecules polymerize to form this actin microfilaments which protrude out in the form of the actin process. This is called as the acrosomal process. So, there are tubules which join together and give rise to this acrosomal process. So, as the sperm is gaining the entry into the jelly layer, these hydrolytic enzymes are released, and this acrosomal process is also enlarged. So, acrosomal reaction is initiated by the interactions of the sperm cell membrane with a specific complex sugar in the egg jelly. So, now again recognition is important. So, acrosomal reaction will not take place until and unless this acrosomal tip of the sperm recognizes certain carbohydrate receptors which are present on the egg jelly, or special complex sugars which are present in the egg jelly. These polysaccharides bind to specific receptors on the sperm plasma membrane directly above the acrosomal vesicle. The egg jelly factors, highly specific to each species, and egg jelly carbohydrates from one species fail to activate the acrosomal reaction even in the closely related species. So, activation of the acrosomal reaction is again one step, or it ensures that there is the species specific fertilization that needs to occur. The binding of the carbohydrate to receptor activates sperm membrane proteins such as a calcium transport channel that allows calcium to enter the sperm head, a sodium hydrogen exchanger that pumps sodium ions into and hydrogen ions out of the sperm head, and a phospholipase enzyme that makes another secondary messenger such as IP3, which releases the calcium ions from inside the sperm. So, binding of the carbohydrate to receptors activate all these proteins that changes the ionic environment inside the sperm. Elevation of calcium ions triggers the fusion of this acrosomal membrane. So, this is the mechanism by which the fusion of the acrosomal membrane and the plasma membrane will take place. The tip of the acrosomal tip of the sperm needs to recognize certain sugars in the egg jelly, and binding to these receptors will cause these proteins to work that will change the ionic environment inside the sperm head, and the change in the ionic environment or the elevation of the calcium ions triggers the fusion of the acrosomal membrane with the adjacent sperm plasma membrane, releasing the enzymes that can lyse a path through egg jelly to the vitelline envelope. Second part of acrosomal reaction involves extension of acrosomal process that arises through the polymerization of globular actin molecules into the actin filaments. So, species specific recognition, the sperm's contact with an egg jelly coat provides the first species specific recognition event. In this regard, the scientist F.R. Lilly have given the fertilizin-antifertilizin reaction, in which he said the hypothesis that in the unfertilized egg, fertilizin molecules are present in the cortical layer of the egg. Each one of them bears a spermophile and a oophile group factor. So, this is the spermophile, this is a oophile group, this is a spermophile group, and these fertilizin molecules are present in the periphery of the egg. When the sperm comes in, there is another molecule called as antifertilizin, and this is the egg receptor which is there. So, this is the egg receptor which is binding here, and this sperm will bind on to the spermophilic site. So, this is the sperm receptor and this is the egg receptor we are talking about. And these are the antifertilizin molecules which are present on the sperm receptors which is there. So, you can see as a result molecules of antifertilizin rapidly block all the spermophile group of the fertilizin molecules, thus preventing the reaction with other spermatozoa. This is Lilly's hypothesis concerning the mechanism of prevention of polyspermy also, because all of them becomes occupied, therefore only one sperm, no more sperms can bind to the egg membrane. Another result of the activation of the fertilizin molecule is the reaction of all oophile group, each with one egg receptor. This is the hypothetical mechanism of activation of the egg. So, the first recognition that happens is the species specific recognition is this fertilizin and antifertilizin reaction that takes place. Thank you. External fertilization poses a problem of species-specific recognition of the gametes. One of the hypotheses given by F.R. Lilly was that there is a reaction between a fertilizin-antifertilizin reaction, that when they contact this, the species-specific recognition occurs, because fertilizin which is present in the periphery of the egg recognizes only a particular type of antifertilizin or a particular type of sperm receptors which are coming. Then another critical species specific binding event occurs once the sea urchin sperm has penetrated the jelly and the acrosomal process of the sperm contacts the surface of the egg. The acrosomal protein mediating this recognition in sea urchin is called as binding. The bindin is capable of binding to the degelid eggs of the same species, causing agglutination of the eggs, but not to the other closely related species, as it is shown in this experiment by Glabe and Vacquer. What they did was they took two species of the sea urchin, and then they saw that in one of the species, when they are putting the bindin particles of the same species, then the agglutination or the clumping of the egg is occurring. However, the bindin molecules from a different different species and egg from the different species causes no clumping of the egg molecules. And you can see here, this is an original picture where the acrosomal process is in contact with the microvilli from the egg, and this recognition is specifically done or mediated by these proteins which are termed as the bindin. So, this is another species specific binding event which makes sure that only the species specific fertilization takes place. Moy and Vacquer in 1979, they also demonstrated that binding is located specifically on the acrosomal process. So, what they did was they took acrosomal process and then they incubated it. It was an immunological technique. They incubated with the rabbit antibodies against bindin. So, rabbit antibodies will bind to the bindin molecules, and then they create a wash so that unbound antibodies are washed away, and then they again put it with the secondary antibody which is raised in the swine, anti-rabbit antibody, which is conjugated with the peroxidase enzyme. When this peroxidase enzyme, then again it is washed, so you get this kind of a complex, and this peroxidase enzymes catalyze the conversion of this diaminobenzidine in the presence of peroxidase. It should be H2O2. In the presence of H2O2, it creates a kind of a deeper precipitate. So, these are the original pictures where you can see the location of the binding or the localization of the binding is on the acrosomal process. This is again in the vitelline membrane. This is an interaction with the vitelline membrane of the egg. This is the acrosomal process, and this is where the binding is located. So, the presence of binding again causes the species specific recognition of the sperm. Vacquer and Payne in 1923 suggested the presence of species specific binding receptors on the egg vitelline envelope. So, they said or they demonstrated that it is not that the binding molecules can bind anywhere on the egg surface, because when they overcrowded an egg with a sperm, they still saw that only approximately 1500 sperms were bound to the egg membrane at different sites, but still the certain places were left. So, the places that were left did not contain the binding receptors. So, the binding receptors are particularly present at certain sites. EBR1 is one such receptor. It is a 350 kilodalton glycoprotein, and it displays the properties of binding receptor, and it has been isolated. So, you can see when the egg is overcrowded with the sperm, still there are certain spaces which are left. And these binding receptors, they form clustered. These are the clustered beads where the sperm can actually bind. So, they are not distributed on the entire surface, but they are clustered at certain sites. So, this binding again provides a species specific recognition which is there. Now, this was all about the action of the sperm. When the sperm is coming and binding to the reaching the egg. So, we saw the steps that how the sperm is guided, particularly in external fertilization, to reach the egg, which is generally by a chemical stimuli. So, they are given direction, they are activated, they are given movement, they are given motive force, and then the sperm reaches the egg. The first thing it does is it comes into contact with the egg jelly layer. So, it releases certain enzymes and certain acrosomal processes to penetrate through this jelly layer. Once it penetrates this jelly layer, so the first species specific recognition occurs at this point, because the jelly layer contains certain proteins which are which are specific for the sperm that is coming. So, that is the first step where the species specific recognition takes place. Once it penetrates through this jelly layer by the aid of the hydrolytic enzymes and the acrosomal process, finally it reaches the egg plasma membrane. Again the species specific recognition takes place due to the presence of the binding on the acrosomal processes and the receptors on the egg plasma membrane vitelline envelope which is there. So, the egg vitelline envelope, again there is a species specific interaction that takes place, and finally the fusion of the two membranes takes place. So, this is from the sperm side. Now, we will look at the how it happens from the egg side or the reaction of the egg, primarily which involves the cortical reaction. Now, directly beneath the sea urchin egg plasma membrane, there are about 15,000 cortical granules, each about 1 micrometer in diameter. Upon the sperm entry, so we have already seen these cortical granules are similar to what the acrosomal vesicle of the sperm was there. It contains again hydrolytic enzymes which are present, and upon the sperm entry, these cortical granules fuse with the egg plasma membrane and release their contents into the space between the plasma membrane and the fibrous mat of the vitelline envelope proteins which is there. First, so there are different enzymes. When we talked about the structure of the gametes, we also talked about the structure of these cortical granules, and there were 4 important protein sets of proteins which were present in the cortical granules, and we will see that they have specific functions during the fertilization. So, this cortical granule membrane fusion with the egg membrane releases its contents into the space between the perivitelline space, the space between the vitelline envelope and the egg plasma membrane, and the enzymes that are released, the functions of those enzymes are like these. The first enzymes to be released are the proteases. The proteases dissolve the protein posts that connect the vitelline envelope proteins to the cell membrane, and they also clip off the binding receptor and any sperm attached to it. So, we will see that when we will talk about the organisms in which the monospermy is the trend, then they have to avoid polyspermy. Usually the sperms are larger in numbers and egg is only a single egg. So, when the larger number of sperms are attaching to the sites, the binding sites on the sperm, but it ensures that the fusion only takes place with the one of the sperm. So, it prevents polyspermy. And to prevent polyspermy, the first step is when these proteases are released, they basically clip off all the binding receptors and their binding sperm. So, one of the sperm, when the successful fusion has taken place, the rest of the binding receptors will just be thrown off along with any sperms binding to that to ensure that there is only a single sperm that has fused with the egg. So, the enzymes released are the proteases from the cortical granules, and these proteases also causes the degradation of the protein posts which are actually connecting or keeping in place the vitelline envelope and the egg plasma membrane. The next enzymes that are released are the mucopolysaccharides. These mucopolysaccharides released by the cortical granules produce an osmotic gradient that causes water to rush into the space between the plasma membrane and the vitelline envelope, causing the envelope to expand and become the fertilization envelope. So, this is a picture which is showing this is an unfertilized egg, and as soon as the egg is fertilized, this is the formation. So, the space is being created. You can see a lot of sperms are bound on the entire surface, but when one sperm has been successful in causing fusing with the egg plasma membrane, there is a space which is created from one side, and this space expands. So, this is the fertilization. This was the vitelline envelope, and this is the fertilization envelope, and this fertilization envelope it's a little raised one, and this happens because of this accumulation of the water that rushes because of the release of these mucopolysaccharides from the cortical granules. And as such, you can see the difference between the unfertilized and the fertilized egg. There is a fertilization envelope that has been formed. Third protein released by the cortical granules is a peroxidase enzyme. This peroxidase enzyme hardens the fertilization envelope by cross-linking the tyrosine residues on the adjacent proteins. So, not only this fertilization envelope is formed, when these proteases are released, it is separating these two membranes, as well as it is just throwing off any binding receptors along with the sperms which is present on the egg membrane, so that only one fusion has taken place. Fertilization envelope is formed and this fertilization envelope is then hardened. The fourth cortical granule protein is termed as the hyaline. It forms a coating around the egg. So the fertilization envelope hardens because of the mucopolysaccharide, because of the enzymes which are basically released, because of these peroxidase enzymes that are released, and apart from that, this hyaline also provide a very much stability or rigidity to the fertilization envelope. The egg extends elongated microvilli whose tips attach to this hyaline layer. This layer provides support for the blastomeres during cleavage. So after fertilization, the next step is that this unicellular zygote will start dividing. So when the cleavage starts, when one cell becomes two and so on, then these blastomeres are kept in place because of this hyaline layer which is there. So this is again a very schematic diagram which is showing the cortical reaction that occurs from the reaction from the egg. So you can see these are all the cortical granules, and these cortical granules have been containing different enzymes, and then these are the portions that are shown green and orange. The orange are basically the enzymes and the green is the hyaline which we are talking about. So the hyaline protein which is there. In fact, the refractile properties of the two differ. Therefore, when you see it under the refractile microscope, then basically it shows a denser and a non-denser thing. So you can see this is the electron micrograph. So you can see that there is these concentric circles kind of a thing which is there because of the differences in the refractile properties of the enzymes and the hyaline layer which is there. So these cortical granules are present in the periphery. This is the vitelline envelope which is there. So this is basically the egg plasma membrane and the outer is the vitelline envelope, and this thrown into the microvilli. We have talked about the structure of the egg, and these red colors are the pillar posts. These are the post or the actin filament posts which are basically present, which are keeping it in the upright direction which is basically there. So now the sperm is entering. When the sperm is entering and it starts fusing, you can see that these cortical granules are now fused with the vitelline envelope and it's with the egg plasma membrane, and it has released its content into the space in between. So this has now raised a little bit. So this now is called as the fertilization envelope, and as you can see, multiple sperms were binding, but when one sperm has already entered and given rise to this kind of a thing, all the other sperms have been thrown out. And this particular vitelline envelope has now been raised and entire water content is there, and this forms the fertilization envelope. So the fertilization envelope now has these hyaline layer which is also present, and these actin protein which was present again become larger and grows into the kind of the actin filaments which is there. So this is a cortical granule reaction. We are right now talking about this cortical granule reaction in the context of the reaction of the egg, but this is a very important step in preventing the polyspermy also, because as you can see, when one of the sperm has managed to cause the fertilization reaction to occur, the rest of the sperms are just thrown out. They can no longer be bound to the fertilization envelope that is formed. So the conversion of the vitelline envelope to the fertilization envelope ensures again that the only one sperms enter the egg and not the multiple sperm, or the polyspermy does not occur. So this is how this is happening. The next aspect is when there has been an action from the egg and there has been a reaction from the, when there has been action from the sperm and there has been a reaction of the egg, then the fusion of the egg and the sperm plasma membranes takes place. So the sperm egg fusion causes the polymerization of the actin in the egg to form what is called as the fertilization cone. Now you can see again there is a homology in the sperm and the egg structures, that actin from the gamete forms a connection that widens the cytoplasmic bridge. So the acrosomal process was formed by the actin filaments, and we have also seen in the egg there were actin microfilaments that are present. So these actin filaments kind of form a bridge through which the sperm nucleus and tail can pass through. So, it forms a cytoplasmic bridge. So, this is the sperm, these are the different microvilli of the egg from where it is entering. So, this is the fertilization cone that is formed, and the sperm fusion, the sperm has started entering. This is when the sperm is actually taken inside the egg, and this is again a section in which it is shown that this is the fertilization cone that is formed, and the sperm with its nucleus and the middle piece, it is completely taken inside the egg which is there. Another important aspect in this case when we talk about is that the fertilization does not just involve the fusion of the two nuclei. It does involve the mixing of we talked about the the purpose or the accomplishments of the fertilization. The first accomplishment was the fusion of the genes or the fusion of the genomes, or basically that the genetic material from the two parent organisms need to come together and lie in a single cell. The second aspect which the fertilization accomplishes is the development of an organism. So, this fertilization is not just a passive process, or egg is also not passively just participating in the fertilization, that the sperm will come and the nucleus, it will put its nucleus inside the egg, the nuclei will fuse and the genetic mixing will be there. It is much more a dynamic process as far as the egg is concerned. So, the mature sea urchin egg is usually a metabolically sluggish cell that is activated by sperm. So, the activation of egg metabolism is an important phenomena or an important accomplishment that is the result of the fertilization, which will cause further development to take place. So, just the fusion of the two nuclei will not solve any purpose, but the egg metabolism needs to be reactivated so that the development into a new organism can occur. Now, the activation of the egg metabolism, there are certain responses which happens within seconds of the sperm and egg fusion, and there are certain functions that happen after a little while. So, there are early responses and there are late responses. So, looking at the events of the early responses, so we will see this is the approximate time post insemination when the sperm has entered inside the body, or basically when the sperm is released into the environment and the sperm and egg contact has taken place. So, it is the 0 seconds when the sperm and egg is binding. So, as soon as the sperm and egg bind, the early response is that within 1 second the fertilization potential will rise. We will talk in detail about these two processes, although we have already talked about the cortical vesicle exocytosis, but we will talk in context of polyspermy in a different session. Right now what we are talking about is that because of the influx of the sodium ions, this fertilization potential will rise. This fertilization rise in the fertilization potential will cause the sperm egg membrane to fuse. In fact, the change in the ionic potential will causes the influx of the calcium ions also. So, calcium ions, there are two mechanisms by which the inner level of the calcium ions are increased. One thing is the influx, opening of the calcium gated ion channels and influx of the calcium ions, and second is that the calcium contained inside the cell is also released. So, the calcium levels inside the cell immediately after fertilization it increases. So, this change in the electric potential causes the change in the plasma membrane which is there, and therefore it will prevent polyspermy, and it will also elevate the levels of the calcium that will cause the sperm egg membrane fusion to take place. The calcium increase is first detected within 6 seconds and the cortical vesicle exocytosis takes place. So, the cortical granule fusion and the releasing of its contents will take place. So, calcium ions, they play a lot of role, a very important role in particularly in fertilization. It not only causes the fertilization, it does not just causes the prevention of polyspermy, but it also causes the reactivation of the protein synthesis, etc. Then you can see, this flow chart explains the function of the calcium release and how it is released and what are its downstream effects, and these are all early responses. The sperm is binding and or fusion to the egg cell membrane takes place, and immediately there is a sodium influx. This sodium influx will cause the membrane potential change, and this we'll see in the context that it will decrease, there will be a fast block to the polyspermy that will occur. Then you can see that there is a tyrosine kinase stimulation which is there. It's a little question mark is here, but there are evidences that tyrosine kinase stimulation is there. There is phospholipase C activation, and there is a IP3 production that also causes the calcium release. Then there is diacylglycerol production causing the activation of protein kinase C, and further causing the sodium hydrogen exchange, which causes the increase in the intracellular pH. So this calcium release will cause one important thing, it causes the activation of this NAD plus kinase, and this NAD plus kinase will cause the conversion of NAD plus to NADP plus. So this is also one of the important thing that occur. Cortical granule exocytosis, we have already seen, it causes the formation of the fertilization envelope with the hyaline layer, and this also causes a slow block to the polyspermy. So the change in the membrane potential will immediately throw off all the sperms that are attached to the egg membrane. Still, if there are some more which are attached, there will be a slow block to the polyspermy, and after the formation of the fertilization envelope, no other sperm can penetrate the egg. The calcium release also stimulates the protein synthesis, DNA replication, and cytoplasmic movements of the morphogenetic material. This calcium release has a later effect also, which causes the increase in the late responses. So you can see in this late responses that there is activation of NAD kinase which is there, which starts at 1 minute. There is increase in NADH and NADPH. This basically alters the lipid metabolism of the cell that is taking place. There is higher consumption of oxygen that is started. So there is a respiration burst that occurs. So there is oxygen consumption that increases. The sperm has now entered, and there is an acid efflux that occurs because the hydrogen ion intracellular hydrogen ion concentration is increased, which means there is increase in pH, and this increase in pH remains high. Then there is sperm chromatin decondensation that occurs. So this is again a very important aspect. When the sperm nucleus enters into the egg, it converts into what is called as the sperm pronucleus. We will also see further down. This sperm pronucleus, because the nuclear membrane, it vesiculates, and it basically becomes open, and the highly condensed sperm genetic content then becomes condensed because the proteins which caused this condensation of the sperm genetic content is exchanged by the proteins from the egg cytoplasm. So the vesicularization of the nuclear membrane makes the genetic content of the sperm to lie in the egg cytoplasm, and this in the egg cytoplasm, the proteins of the DNA of the sperm DNA is exchanged by the proteins of the cytoplasm that causes the decondensation of the chromatin. So decondensation of the chromatin means activation of the transcription from the sperm genetic content. So the sperm chromatin decondensation occurs, the sperm nucleus migration to the egg center occurs where the actual fusion will take place. Egg nucleus also migrate to the sperm nucleus. There is activation of protein synthesis, there is activation of amino acid transport, there is initiation of DNA synthesis that starts. The mitosis begins in around 80 minutes, and then 85 to 95 minutes the first cleavage occurs. So fertilization, whatever molecular mechanisms occur at the time of fertilization, ensures that the zygote is now set on the path of the development into a new organism. So all the reactions, all the changes that is occurring inside the zygote, which is formed by the fusion of the sperm and the egg, will lead to the onset of the formation of the new organism, for which the first step is the conversion of the unicellularity into multicellularity or cleavage. So after the fertilization, the cleavage starts. So the entire activation of the egg takes place, or the egg metabolism takes place, that will give rise to the all different kind of responses leading to this kind of a development to continue. The fusion of the genetic material, the sperm nucleus enters the egg perpendicular to the egg surface. Along with its centriole, the sperm nucleus separate from the flagellum and the mitochondria. So, looking at the early responses of the egg and the late responses of the egg, now we are looking at that how the fusion of the genetic material will take place. So, sperm nucleus is entered, and it enters along with the mid piece which contains the mitochondria and the tail, but as soon as it enters into the egg, it separates from the mitochondria and the tail. The mitochondria and the tail is degenerated, and that is why we say that all the mitochondrial DNA which is coming to the organism is from the maternal side. There is no mitochondria of the sperm that remains. However, the centriole remains, and it leads to the formation of the spindle for the first cleavage to occur. So, the sperm nucleus and the centriole is separated from the mitochondria and the tail, and the mitochondria and tail are degenerated. The sperm's mitochondria and flagellum disintegrate. Inside the egg, the sperm nucleus undergoes a dramatic transformation as it decondenses to form the haploid pronucleus. The male pronucleus rotates 180 degrees so that the sperm centriole is between the sperm pronucleus and the egg pronucleus. So, we call this as pronucleus. We have just seen the sperm pronucleus different from the sperm nucleus because this is more vesiculated. The chromatin has now condensed. It is a little larger in size, and as soon as it separates out, it moves 180 degrees Celsius. So, now there is a sperm nucleus, there is egg nucleus. So, you can see this is a sperm pronucleus, this is the egg pronucleus, and then this is the intramolecular bridge that is kind of formed. So, the sperm has entered through this side, and this is the nucleus which is there, but then it moves to the center, and the egg pronucleus is also moving towards the center. This figure is showing the Ester Formation which is there. The sperm centriole act as microtubule organizing center, extending its own microtubules and integrating them with egg microtubules to form an ester. The microtubules extend throughout the egg and contact the female pronucleus, at which point the two pronuclei migrates towards each other. So, these centriole basically creates a kind of a microtubular organizers. They are the organizing centers, and they will create this network of microtubules on which the egg and the sperm. So, sperm will move, the egg will lie there, and they will finally come together and migrate towards each other. The final thing of fertilization, the fusion from the diploid zygote nucleus. So, the fusion of the two nuclei will take place and will give rise to the diploid egg zygote nucleus which will there, and commencing the next step, which is the mitotic divisions that will occur. So, this is the summary of the events that occurs in the fertilization. This is basically towards the sperm side. As we have seen, the sperm comes in contact with the jelly layer, acrosomal vesicle is released, releasing the hydrolytic enzymes. The acrosomal process is formed, it comes into contact with the plasma membrane, it invades this vitelline membrane. The first recognition, the fertilizing anti-fertilizing reaction happens here. The bindin and the bindin receptors reaction happens here, ensuring that there is a sperm specific interaction, and in fact this acrosomal reaction is activated only when there is a interaction between these complex sugars which are present in the jelly layer with the tip of the sperm which is also there. So, sperm activation as well as the interaction will ensure that there is a species specific recognition. This is the fusion of the vitelline envelope, then it will reach the plasma membrane. Here you can see this is the actin filament which is coming here. The actin filaments from the egg will also come. The cortical granules will fuse. The cortical granules will again release its content in this white space. The fertilization cone will be formed. This white space will be elevated, filled with water, converted into the fertilization envelope, and therefore this through this cytoplasmic bridge, the nucleus, the midpiece and the tail will be engulfed inside the egg plasma membrane. There this nucleus and centriole will separate out and they will migrate towards the center. The female pronucleus, the nucleus will convert into pronucleus by vesicularization and the formation of the decondensation of the chromatin, and finally the two nuclei will fuse together. apart from the fusion of the two nuclei, the egg metabolism will be activated, setting the zygote to the on the path of the development to create a new organism. So, this is all about the external fertilization that we have talked about today, and in our next session we will talk about the internal fertilization that happens in the mammalian cases.
With this note, thank you, ma'am. Thank you so very much for giving us a productive session on external fertilization. Dear friends, we would be meeting again soon to discuss internal fertilization. Till then, we would like you to see the lecture, as we are going to upload the lecture on YouTube very soon, and afterwards, if you feel so that you need to get an answer of any of your questions, then do write to us at info.cec@nic.in. So, with this note, we would like to thank Dr. Charu Dogra Raghavat once again. Thank you, ma'am. Thank you once again.